My name is CheyennePetrino, and for the past 6 weeks, I have been working in the Developmental Biology Lab of Professor Cynthia A. Bradham, under the guidance of graduate researcher Alexandra Lyon. Our goal in this research was to investigate the skeletal and morphological defects that arise as a result of bisphenol A. Bisphenol A, or as it is more commonly known as BPA, is a harmful chemical that was once found in many plastics in the United States. Since then, we have greatly reduced the amount of this chemical in our plastics, but it is still prevalent in many other countries around the world today. This plastic finds our way into our oceans, and therefore so does the BPA. This BPA can have detrimental effects on both organisms and ecosystems. Because of this, it is vital that we understand what BPA is and what its capabilities are. In our research, Alexandra and I performed a wide array of experiments in order to gain a wide range of data from which we could draw conclusions. Some of these experiments include BPA dosing, in which we dose the embryos with diluted BPA in different concentrations, and then we monitor the embryo's growth and see whether it is normal, inhibited, delayed, or whether other defects occur. And what we saw from that is that many defects did occur, and in some cases, development was stunted completely. Otherwise, we did a PMC stain in which we stain the cells that secrete the skeletons of the embryos. Therefore, we can see if these cells are developing properly, and then maybe that is the cause of why the skeleton isn't developing, because if we don't have the skeletal cells, there's going to be no skeleton. So from this data, what we concluded is that there is a range somewhere between 4.5 and 7.5 micromolar BPA that causes these defects in the embryos without resulting in death. Anything higher and the embryos will not grow. Our research is important because it can be applied in many other ways. The species of sea urchin we use, Lidocynus variegatus, is a model organism. Its genes are very highly conserved, and what that means is that its genes are seen in many other organisms throughout our evolutionary tree. So because their genes are highly conserved, they can then be applied to human genes, other animal genes, and so our research can actually be used in the future for humans and other larger mammals, etc. Also, our research can be used as a baseline for knowledge, and it can be used in future research to find cures against these defects caused by BPA.